In this video, we will discuss about if this knockdown or differentiation markers occurs, it will affect the level of differentiation inductions on those cells. So here the differentiation that has been tested is towards the macrophages of this UL60 cells. So first they check whether the differentiations is occurring in the normal cells or not. So they check the two of the important markers for these HL60 cells, that is CD11B and fMLC, and they can see that in the case of the inductions of the differentiations, there is a upregulations of the CD11B and fMLF cells that can be seen here in the flow cytometry results in the panel A compared to the undifferentiated cells, uninduced HL60 cells. Thereafter, what it is, they knock down this guide RNAs. They made a stable knockdown of the guide RNAs of these differentiations markers, that is SPL1 and CBPE. These are two positive differentiations. So these are the two guide RNAs that target the differentiation markers for the macrophages. And for the other controls or other experiments, they target the guide RNAs of this mTOR1 and FLCN. These are not the differentiation markers. So first, from this PCA plot, they checked whether the higher differentiations occurs in the case where differentiation markers is knocked down, or in the case where differentiation markers are not knocked down. So what they are able to find out, that in the case where differentiation markers are knocked down, that is SPL1 and CBPE, they see a lower upregulations of the CD11B and fMLF, whereas compared to the case where the FLNCN and mTOR1 are knocked down, which is not a differentiation markers, they see a high level of differentiation inductions in those stable guide RNA cell lines. And then they check the mean along the principal component analysis along different guide RNAs knockdown, and they are able to find out that in the case with the SPL1 knockdown and CBPE knockdown, the differentiations inductions level is low, because these are the markers for the differentiations and these are knocked down. Whereas in the case where differentiation markers are not knocked down, that is FLCN and mTOR1, they see a high level of differentiation inductions. Thereafter, to check whether they have the different types of differentiations, what it is, they do the differentiations and check the RNA-seq analysis at different time intervals. That is at day 1, day 5 and day 7, they extract the RNA and do the RNA-seq analysis for this different knockdown. We control FLNCN, mTOR1 and SPL1. SPL1 is the differentiation markers. What they are able to find out is that compared to the control, which has minimum differentiations, the mTOR1 and FLNCN differentiated in a different directions and SPL1 differentiated in a different directions. So this further substantiate their point that knocking down of differentiation markers will affect the level of differentiation inductions. Then they also check the average speed, different speed, or the migration speed of this kind of knockdown cells, and they are able to find out in the case of average speed they do not see any significant difference, but for the angular bias, the degree of angular biasness in the form of degree, in the unit of degree, they see significant changes, that is compared to the control. They have seen for the highly differentiated cells, they are able to see a decrease in the angular biasness, and also for the directed speed are also decreased compared to the control. And this all indicates towards that they cells behave different, cells morphologies are or properties are different as they differentiate into the committed cell lineage. In this case, they are checking the differentiations towards macrophages. So this is what they are able to find out, that differentiation markers plays a role towards differentiations of a particular cell lines to a mature committed cell lineage. If you need to understand more about this experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.